All right. Well, welcome to the session on linking cancer cell state to function. I'm Katie Galloway, and I'm really delighted to get to introduce our first speaker. We have Dr. Cédric Blanpain, my apologies, who will be telling us about mechanisms regulating tumor transition states. Dr. Blanpain's group uses lineage tracing approaches to study the role of stem cells during development, homeostasis, and cancer. His group has uncovered the existence of stem cells and progenitors acting during homeostasis and repair of the epidermis, and uncovered a novel paradigm of lineage segregation in the mammary gland and prostate. His lab has pioneered the use of mouse genetics, which I think will be a lot of interest to everyone here, to identify the cells of origin of epithelial cancers and mechanisms regulating early steps of tumor initiation in skin basal cell carcinoma, skin squamous cell carcinoma, and mammary tumors. Additionally, his lab has developed novel approaches to unravel tumor heterogeneity, which is the topic of today's symposium. And what I think is particularly interesting is thinking about how this gives us a better picture of understanding the mechanisms regulating tumor states and how those states contribute to tumor growth, metastasis, and resistance to anti-cancer therapy. And so without further ado, Dr. Blanpain.
Thank you very much for the kind introduction. I would like to thank Matt, Alex, and all the other organizers to invite me. You know, it's a great pleasure for me to be here. Yes. Maybe here in the pocket. No, it's plugged in. Or maybe I can use this one.
You can probably turn it up there.
Can you hear me better? Not much. Yeah?
Yeah.
Okay, perfect. So, as I said, I would like to thank the organizer for kindly inviting me here. It's a great pleasure and a great honor to be here. MIT is the home of my scientific heroes and the people that influence my research in the day-to-day basis and that I admire enormously. So, with that, I will tell you a little bit what I want to discuss with you today. It's what are the mechanisms that regulating tumor states and how this mechanism regulate tumor heterogeneity, metastasis, and resistance to therapy. As you all know, within a given tumor, not all cells are equal. At a given time point, some cells proliferate, others differentiate, some cells invade the tissue, escape the primary tumor, and lead to metastasis, whereas upon therapy, some cells resist to the treatment and can be responsible for tumor relapse after therapy. And these differences between the different tumor cells within a given tumor is called intratumor heterogeneity, and has been one of the key questions that my lab is trying to pursue. So I will illustrate my talk today in one model of squamous cell carcinoma. So the squamous cell carcinoma is the second most frequent skin cancer. It affects about 1 million of patients per year throughout the world. And in most of the cases, it can be cured by surgical excision. Nevertheless, in about a few percent of the case, it can lead to metastasis and be associated with poor prognosis. But more importantly, squamous cell carcinoma is the leading cancer of oral cavity, head and neck, esophagus, and a fraction of non-small cell carcinoma, in which the prognosis is much poorer. And as you will see in my talk, the mechanism that drives squamous cell carcinoma is very similar across these different tissues. And so we believe by studying skin squamous cell carcinoma, we can get insight on a broader pictures of the squamous cell carcinoma and squamous tumor in general. The squamous cell carcinoma, at least the one in the skin, are mediated by mutation in the RAS pathway and P53, and it can come with mutation in HRAS, KRAS, and more recently, another RAS member, RAS2. And it's almost invariably the same type of mutation that arise and that are a driver of these tumors. And so one of the first questions that my group is trying to understand in squamous cell carcinoma was their cellular origin. Is all cells within the skin epidermis is equally sensitive to initiate tumor formation upon this different oncogenic hit. And we found that, interestingly, only the stem cell and not the transient amplifying cells were the cell of origin of the skin squamous cell carcinoma. And there were two types of stem cells, the one that are located in the interfollicular epidermis, which is the part of the tissue that forms the skin barrier, and other cell type, stem cells that are residing in the hair follicle lineage and express LGF5. And however, how these different stem cells and what are the impact of these different stem cells on the tumor heterogeneity of this tumor were not really well understood. And so one of the first things we did was to activate KRAS G12D expression together with P53 in these two different compartments, the interfollicular epidermis and the hair follicle stem cell and its progeny. And to a great surprise, we found that the mutation in the interfollicular epidermis leads to the very classical squamous cell carcinoma. Any pathologist in the world would see this HNE and immediately see this form of keratin pulse, and this would say, you know, this is a well-differentiated squamous cell carcinoma. And when you stain the tumor cells with YFP, here used through the lineage tracing, and K14, an epithelial marker, all tumor cells are K14 positive, whereas vimentin, a marker of the mesenchymal cells, the stromal cells, the fibroblasts are, not staining the tumor cells. But in hair follicle stem cells, most of the tumor that arise was what we call a mixed tumor. A part of the tumor presents an epithelial characteristic here by the expression of K14, but some part of the tumor cells lost K14 expression and gain vimentin expression. And this is a hallmark of what we call epithelial to mesenchymal transition. And I'm sure you know that Bob Weinberg here was one of the fathers of identifying the role of epithelial to mesenchymal transition in cancer and show with many elegant studies that this epithelial to mesenchymal transition was important for metastasis and resistance to therapy. And so we wanted to understand further, why those hair follicle stem cells would undergo EMT upon oncogenic transformation. And so when you stain the YFP cells with an epithelial marker such as EpCAM, but you could have done that with ecadherin as well, you see this nice expression of EpCAM in a monomodal expression in the well-differentiated tumor cells. But in the EMT tumor cell, you see this bimodal expression of EpCAM. The epithelial part and the mesenchymal part are there. And you see here, that's about several hundred of tumors that we have analyzed. And in the hair follicle stem cells, most of the tumor present to some extent epithelial to mesenchymal transition. It can be a little fraction of the tumor, and it can be a complete tumor that are only mesenchymal. And so we try to characterize in detail why these two epithelial compartments were leading to either the well-differentiated tumor or this mixed SCC with EMT. And when we realized that actually the tumor cells co-opt the transcription factor that are expressed in the cell of origin and hijack this program to form this EMT tumor. And the hair follicle stem cell were primed to undergo EMT, and a lot of their chromatin region that allow the expression of EMT genes were already open in the hair follicle stem cells. Whereas the interfollicular epidermis were primed to undergo this squamous terminal differentiation with all the transcription factors that mediate terminal differentiation of the epidermis into the squamous pair were already expressed and primed in this tumor cell. So the second question that we try to investigate was whether EMT occur in the binary fashion, which the profile of EpCAM here would suggest. You see here you don't have a broad distribution of EpCAM. You have really a two peaks that suggest that there was a kind of a switch here that occurred. But whether in this particular peak of EpCAM, either the positive or the negative, we could not know whether the cells would undergo a discrete cell-fate transition and they were more than two states. And to address this question, what Eugenia Pastochenko has done in my lab it was before the occurrence of the single-cell transcriptomic. So we use a panel of cell surface antibodies, and we check whether these different antibodies that recognize either the EpCAM positive cell or the EpCAM negative cells, are they homogeneously expressed, suggesting that, within this different population, it would be an homogeneous population, or are they heterogeneously expressed? That would indicate that there's more things to uncover in this fraction of cells. And the data were very clear. In the epithelial states, most of the tumor were expressing an homogeneous expression of the cell surface marker, suggesting that they are relatively homogeneous population, whereas in the EpCAM negative tumor cells, most of the tumor cells present this heterogeneous expression of cell surface marker, such as CD24, CD51, CD106, and so would indicate that within the EpCAM negative, there's more than one population that are expressed. So we begin to use this cell surface marker 2 by 2 and then 3 by 3 until we deconvolute this tumor heterogeneity, and we realized that with 3 cell surface markers in addition to EpCAM, we could deconvolute most of the tumor heterogeneity we found in those skin squamous cell carcinoma. And these 3 cell surface markers were alpha V beta 3 integrin, CD51, CD61, and CD106. And interestingly, these 2 markers, alpha V beta 3 integrin, were previously found by other groups, including David Chires, to be expressed in metastatic cells and what he called cancer stem cell. And CD106 is BCAM1, and that was just been identified by the group of Johan Massaguet to be expressed into some metastasis-initiating cells in some carcinoma. And so we, using these 3 cell surface markers, we can isolate them and test their functional properties. We can profile them and check their molecular properties. And what we realize is at the beginning of the EMT transition, these two cell population, they co-express at the single cell level K14 and vimentin. So at the early beginning, the EMT tumor cell do not completely erase their epithelial marker expression, and they co-express at the single cell level these 2 genetic programs, the epithelial program and the mesenchymal program. And we call these tumor cells hybrid tumor cells. Bob called them quasi-mesenchymal cells, and other people call that partial EMT. All this nomenclature are really referring to the same cell state, which is this hybrid state in between epithelial and mesenchymal. And then along the EMT, progressively, you lose completely the marker of K14 and the epithelial cells, and they are purely mesenchymal, pure EMT state, what we call late EMT state. You will see that in a moment. And so then we tested the metastatic potential of this different fraction. And initially, we were expecting that the more you are EMTs, more you will be metastatic. And it's not at all what we have found. And we have found that actually the most metastatic cells were not the ones that had completely underwent the full EMT program, were the ones that were in this hybrid EMT state between epithelial and mesenchymal state. And since we have found that together at the same time than many other groups, and since this study, I think there's been many papers across all different cancers showing the importance of this hybrid quasi-mesenchymal partial EMT state in mediating metastasis. Another interesting thing that we found in this in uncovering these different EMT states was they were residing in different niches, and you see here you can clearly identify them by their appearance. The pure epithelial cells are, you see, they form nice cell-cell junction. In this early hybrid state, they still maintain cell-cell junction, but not anymore through adherent cell junction, and there's only desmosome and gap junction that are present in this state, and then progressively completely lose cell junction, but still co-expressing K14 and vimentin, and then they are completely fibroblastic-like state. And that's also something that were interesting is in these different niches, there's a different infiltration of stromal cells in these different EMT states. And more you go to the late EMT state, more you have an infiltration of macrophage, inflammatory cells, and blood vessels and lymphatic vessels at the early stage of EMT, which may also explain why they are more metastatic. So what I show you here is EMT is not a binary process. It's undergo through different discrete cell states. None of those states are extremely stable. Some states are a little bit more stable than the other, but they are very plastic. They can still transit from one state to the others. And they present very different function. And so the epithelial states are the more proliferative. The early hybrid and the hybrid state are the more metastatic. And if you check for their invasion properties, the late EMT state are the more invasive one. And so up to now, we were thinking that, EMT were essentially regulated by, let's say, transcriptional epigenetic mechanism that can be regulated by some intrinsic function and some extrinsic function. But it was not really described to whether the type of mutation that the stem cell undergo, whether these mutations can also influence the occurrence of EMT during tumor heterogeneity. And we got interested in FAT1 mutation because, we have sequenced the chemical-induced skin cancer and found that FAT1 was one of the most frequently mutated genes. And when we discovered that, very little was known about FAT1 function. What was known, it was the second most frequented gene in head and neck cancer. It was extremely highly frequently mutated in lung SCC. It was the second most frequented mutation in the oral SCC. And in head and neck, it's always, and like in many other locations, is a stop-gain mutation, suggesting that it acts as a tumor suppressor. But how this cell surface molecule, it's a kind of a cell adhesion molecule, how this can influence the tumor formation was not really known. And so we did a conditional knockout of FAT1 in a skin model that normally only gave rise to well-differentiated SCC. And what we realized that upon FAT1 deletion, first, the tumor were rising way much more rapidly. They were much more invasive. And then they also co-express K14 and vimentin here, this hybrid EMT state. And they were way much more metastatic in the primary tumor and lead to lymph nodes and lung metastasis with a much higher frequency. And so that was, we thought it was particularly interesting, and we tried to understand how that could work. And so we spent another year or two of trying to identify the underlying mechanism by which FAT1 deletion lead to this hybrid EMT state, and by combining proteomic, phosphoproteomic transcriptional activity, we uncovered that FAT1 deletion will lead to the activation of an enzyme here, calmoninin kinase 2, which phosphorylated SRC and YES protein, which lead to the translocation of yapentase to the nucleus, and which will lead to the upregulation of one of the key transcription factors that mediate EMT, which is ZEB1. And at the same time, this calmoninin kinase 2 inactivates EZH2, which lead to the upregulation of SOX2. And SOX2 is a gene that are promoting tumor stemness, but also that promote the epithelial transcription program of the cells. And so this mutation here will leads to this two distinct transcriptional output. And so by identifying, the mechanism, so basically after our work, there's different group that has found that indeed calcium and calmoninin kinase 2 is indeed important for this hybrid EMT cells, and other group in a totally different system found that indeed SRC and YES are, key to mediate the translocation of yapentase to the nucleus and promote this state. So showing that, what we found here has been, it's also found in other biological contexts. But by identifying this molecular mechanism, we tested whether the requirement of SARC could be a therapeutical vulnerabilities in these tumor cells. And indeed, when we treated the FAT1 knockout cells with a SARC inhibitor, these cells were much more sensitive to SARC inhibitor as compared to the wild type cells, whereas the FAT1 mediated tumor were, insensitive, for example, to EGFR inhibitor. So really understanding how a mutated gene can lead to a particular phenotype may give you some hints about some vulnerabilities in this tumor and can be potentially exploited for the personalized cancer therapy. So now I will show you, unpublished data, and so we are still trying, very hard to further understand how exactly the EMT is regulated, and so we hope to still find novel intrinsic regulator and also understand more precisely how the microenvironment can influence these different tumor states and whether we can find, by understanding that, some potential target for therapy. And so we have been doing that with a very talented postdoc and student, Kevin and Andrea, and a very talented bioinformatician, Sebastian and Gabriel. And so we have done a lot of single cell sequencing of this type of tumor, including the tumor cells and their microenvironment, and also some multiomics. I will show you that just after, and to have a good sense of what are these different tumor states that we identify using this monoclonal antibody, but in a more precise manner. And so basically you find by single cells exactly the same state that I show you that we have identified by monoclonal antibody and immunostaining, is this epithelial tumor state that express high level of EpCAM, cadherin-1, ESRP1, and which are negative for vimentin. And then you lose this expression of EpCAM, and you begin to express, you begin to express vimentin here, but you still co-express K14, the epithelial marker, or K5, integrin alpha 6, and some transcription factors such as PTX1. That's what we call early EMT state. And then as EMT progress, K14 is lost, but K8, another keratin gene, is maintained, and some collagen begin to be upregulated in this particular state. And finally, you completely lose any epithelial gene, and you only express very mesenchymal genes such as collagen 3A, asporin, periostin, or PDGFRB. All these genes are, the classical gene that you'll find in cancer-associated fibroblasts. So that's why in our system, if you didn't have the YFP expression to map their cellular origin, you would have taken those cells for cancer-associated fibroblasts. And that's why also sometimes pathologists that had questioned EMT, because it's not easy to differentiate a cell that completely undergone EMT and a cancer-associated fibroblast. And so this is, the classical EMT transcription factors, and you see here that twist1 is clearly together with Z1 the very early EMT transcription factor that initiate this process, and transcription factors such as NAIL1, for example, comes a little bit later in this cascade, and that's what we are trying to understand a little bit better now. So when we have done this single cell ATAC-seq, now we can identify the chromatin region and different enhancers that are specific for all these different states, and we can see their dynamic across this progression during EMT. And I love ATAC-seq because it's very precise. It's the peaks are extremely narrow and allow you to do a very good bioinformatic prediction of their enrichment of transcription factors. And you will see in a second, how these precision enable us to kind of infer the transcriptional network with a relatively high precision. Of course, this is inference and doesn't mean function, and we need after to test the function of this transcription factor, but that's what we are currently doing. And so I'm going to show you here. You see this collagen trier. There's this enhancer, keep increasing with EMT, as well as those one. It's very beautiful and narrow peaks that we can identify there. And for example, periostin, is unique to this late EMT stage and allow you to have a good sense. And so after, as I said, we can compute, what could be the likely transcription factor. So the first thing you can infer is basically the motif. And the motif, usually you look at that and they say, okay, is this transcription factor? Always take that with caution. You need to check for each of the prediction, the real family of transcription factor, because those motifs are not specific within a given family. And so it's by looking at both the expression of a given transcription factor and its motif that we can infer the most likely transcription factor that act at this different stage. So I'm not going to go through all this listed, but this gives us very good statistical confidence on what could be the gene that are regulating these different states. And so that's exactly what Andrea and the biologists are doing in my lab now. It's generated CRISPR-Cas9 deletion of all these transcription factor and gain of function. And so we are trying to now see both first in vitro and then in vivo, which are the transcription factor, what they regulate, what are the set of the gene they regulate at each step of this tumor state transition. And so it's a lot of fun, and I'm sure it will lead to some interesting results. We can also begin to do what is called in silico perturbation with different program where you remove one of these transcription factor and you, in theory, and you see what could be their impact on the whole transcriptome of a tumor cells, and so to prioritize which among those transcription factors are the most likely to influence the overall state of a given cell. And we found that indeed, some of the transcription factor had, in theory, little impact on the overall tumor state. And so we will see now with the real biological data whether those predictions are accurate or not. The good thing is that the PC0 of this computation that is just along the EMT state. So that's also reassuring that, we're going to test their function along the EMT process. And so, this is a cartoon. Of course, this is nothing about reality. And so it's just for us a model to, prioritize our further experiment. So another important part of our work now is trying to define the role of the extrinsic factor coming from the microenvironment in controlling this tumor state. I show you that these different tumor states, they reside in different niches, and they are interact with different cell types across this process. And the neutrophils, for example, are very much expressed in the early epithelial states and the very early hybrid, and they are really depleted during the EMT. Whereas macrophages is the image in mirror. They are not very highly abundant in the epithelial one, and they keep increasing with the EMT. And so so the same thing. So what we are trying to do with our single cell now is try to compute what could be the factor that arise from the neutrophil, the macrophage, and the cancer-associated fibroblast in regulating these different tumor states. And we use for that, many different tools. We are using CellPhoneDB. We are using NicheNet, we are using WeChat, and none of those programs are excellent on their own, and I'm sure there's a lot of room here to improve this computational prediction of ligand pair interaction. One of the problems there is the database are poorly annotated, and so that I think one of the major problems that we face. There's a lot of ligand pair interaction that absolutely do not exist in the nature that are predicted by those software, and so here I'm sure that the computational approach need to be improved, and I'm sure the bioinformatician across the world will come, with better predicting tools. Nevertheless, it already predicts some interesting candidate that we are currently testing and see how these are influencing the different tumor state, their response, their role during metastasis, colonization, and resistance to therapy. Now I will discuss with you, how we can target the EMT state and see whether these can have an impact on the patient treatment. And so when we had the first RNA sequencing of this different EMT state, we begin to check what could be the molecule that are expressed during EMT, and that can be potentially targetable using existing drugs that were already tested in clinical trial. And our attention has been caught by a molecule that's called netrin-1. So netrin-1 is an axonal guidance molecule that had different receptors, including UNC5B, which is a tyrosine kinase, and that was strongly upregulated during EMT. As soon as the cells lose EpCAM expression, netrin-1 and UNC5B were majorly upregulated. And we decided to collaborate with a team in France led by Patrick Mellen and Agnès Bernet, who had developed a therapeutical monoclonal antibodies that block the interaction between netrin-1 and UNC5B. And so Patrick was a good colleague and a friend of mine, so he immediately was excited about this potential collaboration, and he sent us a lot of therapeutical antibodies and a PhD student, Justine Langrand, who stayed in my lab actually the next 4 or 5 years to finish the study and really did an amazing job together with the postdoc in my lab, Genia Pastuchenko, and Sébastien was the bioinformatician working with us on that one. And so netrin-1 is expressed in a broad range of cancer, and it has bin shown that if you block netrin-1, you can decrease the proliferation and you sometimes induce apoptosis of those cancer cells, and that's what led Patrick and his team to develop these therapeutical monoclonal antibodies. So when we treated the mice that present these spontaneous EMT tumors upon KRAS G12D and p53 deletion, we induced a recombination, and then 1 month later, we begin the treatment with anti-netrin-1 antibody every other days. And what we found is that the anti-netrin-1 antibody strongly decreased tumor formation, but more importantly for us, shift the proportion of late EMT tumor cells toward the more epithelial tumors. So it's not a really on and off thing. We don't completely block EMT. We inhibit the process of EMT, and we inhibit the severity of EMT in this tumor. And by decreasing EMT, we decrease metastasis, lung metastasis, either the spontaneous metastasis that arise naturally following the KRAS and p53 deletion, but also when we inject them IV, so suggesting that it really act on the metastatic colonization and metastasis formation. Also, and that probably more importantly for the clinical therapy, the co-administration of cisplatin-5-FU together with blocking Netrin-1 function really strongly decrease or promote the sensitivity of the tumor cells to chemotherapy, and that's probably what was the most important thing for the clinician. So we try to understand how the Netrin-1 antibody indeed restricted EMT. So that was the early single-cell sequencing that we have done. And so that's exactly the same. It's not a different UMAP, but show exactly the same state: epithelial, early hybrid, late hybrid, and late EMT. And when we administrate anti-Netrin-1 antibody, as you see here, there's a major upregulation of this epithelial state and a very strong and a completely disappearance of the late EMT state here, and leaving a very tiny fraction of early and late hybrid EMT in place. So now, how targeting Netrin-1 influence the EMT process? And so the first thing we did was trying to understand whether Netrin-1 acts in a cellular autonomous manner in tumor cells, or whether it does that by acting on the tumor microenvironment, because indeed Netrin-1, in addition to be expressed on the tumor cell, is also expressed on a fraction of cancer-associated fibroblasts. So we isolate tumor cells from the primary tumor, culture them in vitro, and when you culture those tumor cells in vitro, they can undergo EMT in vitro without doing anything. You culture them, they have this spontaneous switch, and that, we isolate first EpCAM positive, we culture them, so we start with 100% EpCAM cells, and you see here, after 15 days, half of the tumor had switched and lost EpCAM expression. And when we did knockdown of UNC5B or knockdown of Netrin-1, we considerably diminish this spontaneous EMT, so supporting the notion that at least Netrin-1 can act in a cellular autonomous manner to control EMT through its interaction with UNC5B. To gain further confidence about this prediction, we did RNA sequencing of the knockdown of UNC5B and Netrin-1, and as you can see here, about more than 50% or about 50% of the transcriptional program are co-regulated, and so the same gene are up or down-regulated by knocking down of Netrin-1 or UNC5B, and many genes of the epithelial state are upregulated following the Netrin or UNC5B knockdown, and many genes of the EMT program are down-regulating following that, supporting that this signaling pathway in tumor cells is important to controlling EMT. We don't, of course, rule out that there's a role of Netrin-1 coming from the microenvironment in further regulating these states, but that will need further experiment. So to which extent what I'm showing you here can have some impact on EMT in human cancer. And so the first thing that we did is to interrogate and see whether there's different gene signature that researcher has developed for the human EMT score. And as you can see here, when we correlate the EMT score with the Netrin-1 or UNC5B expression, actually Netrin-1 is a poor predictor of the EMT score. But the UNC5B, its receptor in tumor cell, is very nicely predicted the EMT score with the different EMT score and in different type of cancer that can present EMT. So suggesting that this signaling through UNC5B can be important in human patient with lung cancer. So then we turn toward a more functional experiment, and we use a cancer cell line that many people are using in the EMT field, in which when you give TGF-beta, these cells rapidly switch from an epithelial-like state to a more mesenchymal-like state. And so we treat it with TGF-beta1, these cells, and as you see here, there was an upregulation of Netrin-1 expression, a loss of cadherin-1. And when we transplant those cells in vivo in immunodeficient mice and keep treating them with this anti-Netrin-1 antibody, we prevent the loss of Kettering-1 expression, supporting this idea that by blocking Netrin-1 and 5B interaction in human cancer, we can decrease EMT in those cell lines. But that's, was, again, cell line. So we had a huge opportunity with the clinical trial that were made by Patrick Mellen in patients in which we had biopsy of patients before and after anti-Netrin-1 treatment in a cohort of patients with endometrial cancer. And so we had access, my lab had access to few patients, and he had done single-cell RNA-seq in order. I'm just showing, the result of our lab here. And what we found when we measured an EMT score, which is the fraction of the tumor cells that express vimentin that were decreased following in 3 to 4 patients, was decreased following anti-Netrin-1 therapy, and that was confirmed in a much higher cohort of patients with single-cell RNA-seq in the lab of Patrick Mellen. So together suggesting that also in patients, anti-Netrin-1 antibody can inhibit EMT formation. And so what they are doing in France, they keep moving with this clinical trial, and now they try to associate it, anti-Netrin-1 antibody with chemotherapy or with immunotherapy, because, and I'm not going to talk about that myself, but the group of Bob Weinberg has also found that in addition to be resistant to chemotherapy, those EMT tumor cells can be also resistant to immunotherapy, and that's why they are trying to see whether by decreasing EMT in those patients, they can respond better to immunotherapy. And so now to finish, I will discuss you, because I didn't touch upon that. I show you that indeed EMT tumor cells are more resistant to chemotherapy, but I didn't tell you at all what could be the mechanism that mediating this resistance. And that's the work of Maud de Bonny, who is an MD and a head and neck surgeon, and that did a PhD in my lab. And she really studied in detail how these EMT tumor cells could resist to therapy. So the first thing, that's the model that you have seen already throughout my talk here. EpCAM positive are the epithelial tumor cell, EpCAM negative are the EMT tumor cell. As you see here, when you give to the mice cisplatin 5-FU and you look 24 hours later, what is the proportion of active caspase 3? And you see that the EMT tumor cells are much more resistant to cisplatin 5-FU induced cell death. Okay, that it's consistent with what already people have shown. It's not only true for in vivo, it's also true in vitro here. That's the cells that are isolated, cultured in vitro, and treated with cisplatin 5-FU. And again here, the EpCAM negative cells are much more resistant. So it's not only true for cisplatin 5-FU, but it's true for a wide range of chemotherapeutical agents. And so we begin to be interested and check what in our transcriptomic data could explain why EpCAM negative EMT tumor cells were resistant to chemotherapy. And what we found, there was a very high expression of a small GTPase called ROJ that was previously identified in melanoma cells to mediate resistance to cisplatin. And the author of this study had proposed that ROJ was regulating resistance to chemotherapy by regulating P53 and SOX10 expression. We knew that, although that it's totally possible to occur in melanoma cells, in our model, since our model all cells are P53 knockout, cannot be mediated by P53 regulation, and SOX10 is a transcription factor that are more expressed in neural crest-derived cells and melanoma cells, but not in epithelials. So we try first to see whether ROJ is indeed an active player in this resistance to chemotherapy. So the first thing we did is using shRNA against ROJ and treat it with chemotherapy. And as you see here, the ROJ knockdown massively increased the cell death following chemotherapy. And so that's using active caspase-3. Here's just by counting the number of cells. We did the reverse experiment. We overexpress ROJ in cells that are normally sensitive to chemotherapy, such as the EpCAM positive cells. And as you see here, the EpCAM positive cells, which are not normally very sensitive to chemotherapy-induced cell death, are now very resistant. So clearly, using gain and loss of function, ROJ regulating the resistance to chemotherapy associated with DLT. We want to be a little bit more precise and did that with the flox allele. So we import the flox allele, cross them with our model, and the knockout of ROJ in primary tumor didn't really change their EMT. There was a decrease of tumor formation though, but in those models, when you also give chemotherapy, the ROJ knockout were much more resistant much more sensitive to chemotherapy-induced cell death as ROJ wild-type cells. And that both in vivo, in vitro, and in long-term in vivo treatment. You see here, the knockout here respond very nicely to chemotherapy, whereas the ROJ wild-type cell keep progressing upon chemotherapy, really demonstrating that ROJ is a key regulator there. So know how ROJ, a small GTPase, is regulating this chemotherapy-induced cell death. And so the first thing we did, transcriptomic, there was not much differences. Then we moved and we reasoned that, indeed DNA, the response to DNA damage, etc., are mostly mediated by protein modification. So we did a proteomic approach, and then the data were really clear. And ROJ knockdown is regulating differential abundance and phosphorylation of many different molecules that are known to be important for DNA origin of replication and the regulation following replication stress. So we test whether indeed ROJ could decrease DNA repair, and so for that we use gamma H2OX, which is a very good marker of unrepaired DNA. And indeed, upon ROJ knockout, there was a much stronger H2X expression. That's a quantification by flow cytometry. It's even much more dramatic. And there was, the ROJ knockout clearly impaired the resistance of the EMT tumor cells to DNA damage. We collaborate with the group of Juan Mendez in CNIO, which is a specialist in the origin of replication and replication stress, and using this double labeling CDU, IDU, which allow you to measure the number of origin of replication and the speed of the DNA replication. We found that the speed of replication was relatively similar across the group, and chemotherapy decreased the speed of replication, as you might expect it, upon DNA damage. But in the EMT tumor cells, in a ROJ-dependent manner, there was an increase in the recruitment of de novo origin of replication, of dormant origin of replication, which allow the cells to form more of these loops that allow DNA repair through homologous recombination. We then finally we perform pull down following by mass spec of the interactor of ROJ. And what we found here, we found that there was a lot of protein that regulated actin polymerization, and including a nuclear actin transporter importin 9. And so we were very intrigued by this finding. And at the time we found that there was two papers that were published in Nature suggesting that nuclear actin can indeed accelerate the DNA repair through, some kind of unknown processes. And so we tested these possibilities, and for that we use a chromo body. So it's a single chain monoclonal antibody that recognizes nuclear actin. And we found that this type of branch nuclear actin was much more abundant in EMT tumor cells in a ROJ-dependent manner. When you knock out ROJ, there's a decrease in this type of cells. And by blocking actin polymerization, we could rescue partially the resistance of EMT tumor cells to chemotherapy. and the recruitment of de novo origin of replication and DNA damage. So altogether, what our data suggests that one of the mechanisms by which EMT tumor cells can resist to chemotherapy, it's by promoting a faster DNA repair. And this faster DNA repair can be mediated through the recruitment of nuclear actin and the formation of de novo origin of replication that help the cells to cope with DNA damage and repair the DNA faster. So with that, I thank our collaborators, Thierry Bout and Sébastien Van Uitven, for the bioinformatics support, Agnès Bernet and Patrick Mellen for the Netrin-1 antibody, Juan Mendes and Sarah Rodriguez for their work on the origin of replication, and Yvan Sires and Steinhardt for bioinformatics support for both the cell-cell communication and gene regulatory networks. And I thank all my fantastic team that work very hard to do their passion and try to advance a little bit our understanding of the tumor state. Thank you very much for your attention. I will be very happy to answer your questions.
Will we have time to question facilities come the mic? Question?
Really impressive talk. I have two questions. The first one is more on the basic molecular mechanisms of how the different populations are spatially organized and reorganized within tumors. Do you think that these programs are being predetermined as the cells initially acquire a genetic lesion, and they are predetermined to become a niche supporting cell or quasi or partial EMT-like supporting cell? And the second one, which I think speaks to the power of mouse models, where if you didn't have the LSL-YFP allele, you wouldn't be able to pull out non-fibroblasts. Do you think that those cells are essentially acting as a CAF? And whether depletion of CAF cells in other models, do you think they actually have been depleting quasi-mesenchymal or partial EMT cells?
Thank you very much for all these very important questions. So they are very different, and I'll try to answer them one by one. So the first one about the spatial organization of the tissue. Indeed, the tumor cell that arise from the hair follicle stem cell undergo rapidly EMT. And so what you see in their spatial localization are more or less their appearance during the time. And you can see that even in a very small tumor, you can already see the first emergence of EMT appearing. And for sure, there's not only the epithelial part that are proliferating. So when you generate the first step of EMT, this early hybrid, they are still proliferating, and so they are not completely devoid of self-renewing properties. That's the first part. So how do they reorganize their niches during tumor regenesis? Their transcriptional programs are associated with the secretion of different cytokine and chemokine that will attract their own niche. And for example, in EMT, you have a lot of molecules that are attract macrophage and other angiogenesis as well. So clearly, they can self-organize their tumor microenvironment by secreting the factors. So that's for the first part. The second part was yes, exactly. So this is also something very important. I think you really point something to that is very critical. In the late EMT stage, when these cells are almost fibroblastic-like, you can differentiate them on using single cell, but they are very similar. Actually, they are very deprived of real cancer-associated fibroblasts. And they indeed probably do the role of cancer-associated fibroblasts themselves. They express all the classical, as you saw, extracellular matrix. That's the #1 gene ontology program that's going on there. They secrete all their supporting microenvironment, and they don't need any more cancer-associated fibroblasts. So that's also very important. Yeah.
Hi, great talk. So I have actually two quick questions. The first one, there were recent reports about P16 overexpression mouse models where hyperplasia that might represent onset of actinic keratosis is also associated with the massive loss of the hair follicle stem cells. Do you think and do you anticipate that these stem cells migrate to the epidermis in order to start the actinic keratosis and later on the squamous cell carcinoma, or whether they're eliminated by the immune system? This was my first question. Second question is actually I'm fascinated by the different states of the EMT in different fields or niches within the cancer. Do you think DNA damage is the major player that recruit immune cells, including macrophages, and not necessarily EMT directly?
So I'm going to try to step in different parts. So the first question about do hair follicle stem cells migrate toward the interfollicular epidermis in the homeostatic condition? The answer is no. So you need some kind of damage or wounding stimuli to promote their migration. And it's still not very well understood why there's these boundaries that compartmentalize the different part of the skin epidermis in subcompartment, but clearly in the absence of damage, each part sustain their own cellular compartment. Upon wounding, indeed, they are migrating. Whether upon oncogenic stimuli they are migrating, RAS alone is not sufficient to make those cells migrating upward. But RAS plus a small wounding stimuli massively recruit those cells toward. So clearly, the oncogenic pathway can promote the cells to exit their initial niche and promote tumor heterogenesis and also increase tumor heterogenesis.
Second question about the DNA damage.
Yeah, DNA damage. We don't see gamma-H2X or any sign of DNA damage to be present in the absence of therapy. Again, it's not because we don't see that at the bulk level by Western blot that, there's some DNA damage, because something that is clear, though, the genetic instability is higher in EMT, in our model, in EMT tumor cells in the epithelium. And a part of the transcriptional program can sometimes, or at least the clustering, can be linked to the copy variation number as well. So clearly, there's something there that we don't fully understand. Is this because there's a change in the mechanism that regulate the aneuploidy or decrease sensing of aneuploidy, and that can lead to cell death? We don't know.
I see. Thank you so much.
My question is kind of regarding your second to last story regarding the EMT transition inhibitors. I was wondering in regard to that if there's any concern to the potential to increase metastasis, I guess. So I guess in the event of preventing further transition to EMT, you are, allowing them to be more sensitive to chemotherapy, but you also showed earlier in the talk that as soon as they decrease EpCAM, they begin this transition, there's this massive spike in metastatic potential. So I guess, did you guys look at potential metastases in the case of this inhibitor, or does it kind of reverse the transition all the way back to epithelial?
You're completely right. This is, and is going to be always a fear in this EMT field as kind of a double hedge. In one hand, you can, sensitize the cells to chemotherapy, but if you may, and we still don't really understand why MET, the reverse process, indeed seems to be associated with lung or metastasis outgrowth. And so the risk would be that by promoting the response to chemotherapy, you would promote the metastatic outgrowth. This is something that we have in mind. Fortunately, and maybe with the Netrin-1, it seems to decrease also metastasis. But I'm not sure that the whole spectrum of anti-metastatic potential of Netrin-1 is only by changing EMT. It can be also by changing the niche and the cellular interaction between the tumor cells and the thing. But you're right that any therapy that will aim at be used in therapy to stimulate response to chemotherapy needs to be balanced by its pro-metastatic potential.
Thank you.
Let's say that is one more time.
Thank you.
And our next speaker is Karuna Ganesh, who's coming from Memorial Sloan Kettering, telling us about dissecting targeting plasticity and metastatic colorectal cancer. And I'm going to let her get started right away. While she's getting that up, we'll just let you know that her lab works on investigating and treating metastatic gastrointestinal cancers. She's an attending physician in oncology and service, a director of metastasis research in the Center for Colorectal Cancer at MSK. Wonderful.
Thank you for the introduction. Can you all hear me?
Yep.
Okay, great. Thank you so much for this wonderful invitation to speak at this symposium today. It's a real honor and privilege to be here. So here are my disclosures. I'm a physician scientist. I'm a medical oncologist treating patients with advanced cancers. And it is for this reason, really, that my lab studies metastasis, because it is metastatic disease that is the cause of the vast majority of cancer death. We're actually, in 2024, pretty good at treating this disease, stage 1 to 3 disease of any sort, from any cancer type, disease that is a cancer but is restricted to the organ where it initiated, a primary tumor. And we treat these tumors by cutting them out. Our surgeons are excellent. But it is once these cancer cells learn to disseminate, spread to distant organs, and eventually grow out into macrometastases. These are clinically apparent as at least 1 cubic centimeter lesions on imaging such as CTs or PET scans. This is really where the problem starts. And this is the disease that in 2024, despite all of the advances, remains largely incurable. And with our best treatments, therapies in 2024 with immunotherapy, targeted therapy, chemotherapy, at best what we can do is debulk these tumors, control them for a while, but from these stem-like states, they then re-emerge time and time again to cause more aggressive disease that eventually takes over organ or organismal function and kills the patient. And so the question that my lab asks is, what's different? What's different from these primary tumors to these metastatic tumors? And of course, seminal work from Bob Weinberg and others following him has shown that these cells that leave the primary tumor to initiate metastases by undergoing EMTs or other cell state transitions are already different from the primary tumor cells from which they started off. But what remains a black box largely is what happens in this overt macrometastatic state. And to ask this question rigorously in patients, ideally we need to compare within individual patients and across cohorts matched primary tumors and metastases cut out of the same patients at the same time to understand how the cells have evolved and how their vulnerabilities have changed as they go through these many rate-limiting steps, these many hurdles of the processes of intravasating into blood vessels, surviving all of the stresses, the immune pressures of being in the circulation, and eventually colonizing a hostile alien niche in the target organ. And so to address this, what I set up at Memorial was a protocol where we could get from patients undergoing surgery for metastatic colorectal cancer. And this is really a unique opportunity because unlike in most solid cancers such as breast or lung cancer, once patients have metastatic disease, the standard of care is not to do surgery. The standard of care is to treat with systemic treatments. But this means that we don't have access to tissue from the metastatic site that we can study. And colorectal cancer provides this unique opportunity to study this because surgery for metastatic disease remains a standard of care for some patients. And so what we were able to do was to collect matched normal colon, primary tumors from the colon, and liver metastases, and in some cases other metastases, cut out synchronously at the same time from the same patients across cohorts. And what we could do is get the fresh tissue, bring it to the lab, sort them into single-cell suspensions to do single-cell transcriptomics on these matched trios of normal, primary, and metastasis, as well as look at the tissue using spatial technologies, including multiplex immunofluorescence. And this allows us to ask the questions about what happens to the cell states within these tumors as they transition to the metastatic state. And importantly, what we also do is from a portion of the same tissue that we, suspension that we use for the single-cell transcriptomics, generate patient-derived organoid models, which then provide us with these genetically tractable model systems that we can use to understand mechanisms using either genetic or pharmacological perturbations. So we've generated a large number of data sets from 31 patients. About a third of these had previously received no treatment prior to their surgery. So this allows us to look at the ground truth of metastasis, and 2/3 had received a standard of care 5-fluorouracil-based chemotherapy prior to their treatment. We also longitudinally follow them so we can get subsequent metastases that occur 1 or 2 years later and look at how the cancers evolve both in space as well as time. So what do these data look like? So I'm showing you today just the epithelial cells, just the cancer cells. And in the intestine, for a long time, it's been known from the work of Hans Klevers and others that the cell of origin of a primary tumor in the intestine is this LGR5 stem cell. And these are the stem cells that are required to maintain homeostasis in the gut. They live at the bases of these intestinal crypts, and these cells divide and differentiate to give rise to all of the other cell types that populate the intestinal epithelium, including the absorptive enterocytes, which absorb nutrients from the gut, shown here in blue, and the secretory goblet cells that secrete mucin into the gut. And indeed, in our normal cells, we can see these two lineages beautifully represented, as well as the intestinal stem cells. And then we see this large maroon mass of cancer cells across these patients. Now, to analyze these data, because this is coming from human tumors, each tumor is different from the next. They have different genomic alterations. And what we're really looking at is not what are the interpatient differences that might be driven by the genomics, but what is driving this conserved, shared biology of metastasis that is common across all of these patients despite their genomic differences. And to do this, we adopted an approach called hotspot. And what hotspot allows us to do is to do factor analysis to look for co-regulated modules of gene expression that are shared across all of the tumor cells in our data set from these 31 patients. And when we do this, we identify immediately a number of co-regulated modules of gene expression that resemble gene programs that we expect to find in the normal intestine. These include intestinal stem cell-like programs, as well as programs that resemble the more differentiated populations of the intestine. And we call these canonical intestinal programs. This is what we expected to find. That's what has been shown in mouse tumors as well as in human primary tumors. But we also found a whole bunch of these new modules that had nothing to do with the normal intestine. And we call these non-canonical. And when we look closer at what's going on here, we start to see programs associated with EMT. We also see programs associated with injury repair, wound healing, programs that we and others have previously shown to be associated with metastasis initiating cells. But the big surprise was that we found these whole bunch of co-regulated modules of gene expression that had that were completely different differentiation programs, but not differentiation associated with the intestine. But now we see evidence of lineage plasticity or turning on of gene programs associated with osteoblast or bone lineage, squamous or skin lineage, and neuroendocrine lineage, something that has not previously been observed in these cancers. So we then were curious to know across patients, what is the distribution of these canonical and non-canonical states? So shown here in the cooler colors, the blues and greens represent the canonical intestinal programs, and the warmer colors in reds and pinks represent the non-canonical programs. And each line represents a patient. So each patient's tumor, as you can see, spans a different place along the spectrum of canonical to non-canonical differentiation. But even in these most canonical tumors, we already, now that we know what we're looking for, we start to see at least a small number of cells that express these non-intestinal, non-canonical programs. And at the other end of the spectrum, there are tumors that are completely non-intestinal in nature. When we look at the matched primary tumors and metastases in our cohort, we see across the board that the metastases are much more likely to be non-canonical, whereas the primary tumor-derived cells are more likely to be canonical. We then asked, does this matter? Does non-canonical transdifferentiation matter? And to do this, we used our hotspot modules of canonical and non-canonical gene expression to generate signatures that we could now use to interrogate much larger clinically annotated cohorts of bulk RNA-seq data sets. And we did this in 2 independent cohorts. One was the TCGA, which is a collection of pre-treatment primary tumors with both bulk RNA-seq as well as clinical outcome annotation, and a second cohort that we had internally of patients with locally advanced rectal cancer, again, pre-treatment primary tumors. And across both cohorts, we found that expression of these non-canonical programs, the warmer colors, in the pre-treatment primary tumors was associated with poor outcomes, both tumor recurrence in both cohorts, as well as, most importantly, overall survival. So if the tumor expressed these non-canonical programs, this was associated with poor outcomes, including poor overall survival. And what is exciting here is that we see that across the board, despite all of the differences across these patients, across their tumors and genetics, we find that metastatic progression shows this convergent evolution with a shared shift in cell fate from canonical intestinal to non-canonical divergent fates. And these non-canonical states are really pervasive across patients, and they portend poor outcomes. We can also see this at the spatial proteomic level. And when we now look at the normal colon in patients, we see the single layer of cells surrounding these intestinal crypts. In the primary tumor, the crypt architecture is lost, the tumors are distorted, but all of these cells still express the yellow CDX2 marker, the intestinal lineage-defining transcription factor. Whereas in the MASH metastases cut out at the same time from the same patient, we see only a subset of these cells still express the CDX2 in yellow, and there are other cells that have lost CDX2 expression that have now gained expression in pink of chromogranin, a neuroendocrine marker, in blue SOX2, a dedifferentiation stemness marker, and these large sheet-like patches of cells that express keratin 5, a squamous skin differentiation marker. How do the cells then switch from canonical state to non-canonical phase? And to do this, we were reminded of the idea that metastasis is effectively a process of regeneration or wound healing gone wrong. And across epithelial cells, tissues, including in the small intestine, there is growing recognition, as Cédric showed, that during wound healing, there is a transient reversion to a high plasticity state. And this high plasticity state often has features that are shared with developmental programs that are present in embryogenesis. So to ask whether this sort of process might be happening during metastasis, we turned to the Human Fetal Gut Atlas, which is a data set generated in the UK from aborted human fetuses from the first and second trimesters, where Sarah Teichmann's lab was able to do single-cell sequencing on these tumors. And we compared the genes that are expressed in the first trimester in the endodermal progenitor cells that will eventually go on to make the colon, to the genes that are expressed in the colon in the fetus in the second trimester, where major organogenesis is complete, and you can see all these differentiated cell populations appear. And by plotting the first and second trimester gene signatures along the axis of differentiation that we find in our data of tumors from canonical intestinal to non-canonical, we see that the second trimester differentiated intestinal score goes down along this axis, suggesting a progressive loss of intestinal lineage identity, whereas the first trimester fetal progenitor score goes up, peaks as an intermediate, and then goes down again at the very end. And when we plot all of our gene programs from the hotspot modules in this way, we now see again that the fetal progenitor score serves as a regenerative intermediate, spanning this trajectory with the intestinal programs at one end, and then entry into this fetal progenitor intermediate, and finally transdifferentiation and lineage plasticity with the gain of the non-intestinal neuroendocrine and squamous programs at the other end. Across our tumors, the primary tumors live largely in these intestinal states, whereas the metastases span the full spectrum of intestinal, fetal, and non-intestinal states. So we then asked, how conserved are not only the gene programs, but the sequence of gene programs across patients? Can we now predict, based on where a tumor is, where it might end up in the future? And to do this, we looked at the overlap in the gene programs, because a single cell in a tumor can express more than one gene program. And we can assign a certain sort of cell state to a cell based on the most prominent gene expression program, but we can also see to what extent do they express other programs. And based on the extent of expression of shared programs, we can create a sort of pseudo-ordering of cell states along this axis. And what we found was that across all of the 31 tumors, patients in our data set, we found actually the exact same ordering of cell states, from differentiated intestinal to intestinal stem cell-like, fetal, and then finally non-canonical. The same sequence was also shared in the second data set that we looked at of 6 additional paired primary MET samples that was published last year. And so what we really find is, which is exciting from a therapeutic standpoint, is that despite all of this heterogeneity, cancer cells display highly conserved, predictable, sequential cell state transitions during cancer progression. So now that we know what we know, we can start to ask mechanistic questions. Is this transdifferentiation something that's happening because of the cancer cells themselves acquiring different epigenetic states during tumor progression? Or is this purely a difference in the microenvironment? Because clearly the niche in the colon is different from that in the liver. So to ask this question, we turn to our organoids. So in this tumor, for example, we found that the primary tumor occupied largely intestinal states. This is a single-cell RNA-seq of the tumor, fresh from the patient. The metastasis spans the full spectrum of states. And now what we do is isolate just the cancer cells and control entirely the microenvironment by growing them in organoid conditions. And we can choose what we put in the organoid media. And so we start off by growing these organoids in standard organoid condition, human intestinal stem cell media, which, as the name suggests, contains all of the growth factors necessary to promote an intestinal stem cell state. And then we did single-cell RNA-seq on the organoids and plotted the probability of each cell in the organoids either adopting an intestinal stem cell-like state at the bottom left, a differentiated intestinal state at the top, or a non-canonical state at the bottom right. And we find that despite the big differences that we saw in vivo, when we now put all of these cells in the same organoid conditions, we find that most of them adopt the intestinal stem cell state. So they actually converge. However, when we withdraw intestine-specific niche factors, which is, of course, what's happening during metastases as these cells leave the niche, we now see something strikingly different. The primary tumor cells remain largely intestinal. They differentiate a bit, but they cannot do anything apart from behave intestinal. Whereas the metastases completely now turn off the intestinal program and turn on these non-canonical programs. So this tells us that cells themselves have become epigenetically different, with the metastases becoming less lineage restricted and much more able to adapt to microenvironmental cues as they evolve. When we transplant these organoids into the cecum of the mouse, both can grow beautifully in this intestinal environment, whereas in the liver, the primary tumor organoids fail to adapt to this environment and peter out, whereas the liver or metastasis-derived organoids are, of course, able to grow in this niche. And when we look at the tumors that we get from these xenograft liver tumors, we see that these organoids in the liver have retained their ability to undergo multi-lineage differentiation, both into the yellow intestinal states as well as into the non-canonical states. Chemotherapy is another form of epithelial injury, and we tested whether this could also promote non-canonical differentiation. And indeed, we found that when we treated both the primary tumor and the metastasis organoids with chemotherapy, we could at least transiently push them into non-canonical states. And with multiple rounds of injury, we can epigenetically fix them in these states. So metastasis and chemotherapy both promote these highly plastic non-canonical cell states through epithelial injury. What are the mechanisms that underlie this plasticity? And the way we asked this question was to ask what factors might restrict intestinal lineage in the primary tumor, and could these then get dysregulated as these tumors progress? And by computational analysis, we identified a transcription factor called PROX1, which we hypothesized could be serving as a transcriptional repressor of non-intestinal fates in the injured epithelial cells. PROX1 is expressed at the invasion front of multiple primary tumors in these cells that are de-differentiating, that are losing CDX2 expression, losing intestinal identity. And indeed, when we tested this hypothesis genetically, when we knocked down PROX1 in the primary tumor organoids, we found that indeed it was acting as a repressor because the knockdown then unleashed the expression of this entire non-canonical program. In contrast, when we did the same experiment in the matched metastasis organoid from the same patient, these organoids are already more non-canonical to begin with, and further inhibition of PROX1 didn't really do very much. So PROX1 function is not really important anymore in metastasis, where the dependency is much weaker. We then looked at this across organoids spanning the full spectrum of canonical to non-canonical states. So of course, multiple factors come together to establish these cell states. And we found that the more canonical the organoid is, the greater the effect of PROX1 in inhibiting the non-canonical programs. Whereas the more non-canonical the organoid is to begin with, the less effective PROX1 is at repressing the non-canonical programs. Of course, we're very interested in understanding the mechanisms by which PROX1 becomes ineffective in metastasis and in non-canonical states, but this is now work in progress. So what we learned from this is that as these cells traverse the Waddington's landscape, to go down different lineages. There are genes like PROX1 that are important in establishing these barriers in the regulatory landscape. And what's happening during metastatic progression is that these barriers fail, and this in turn allows promiscuous expression from different lineages to happen, and allows these metastases to be highly adaptable. So in the last minute, I just want to address this question of how we might think about targeting this complex biology. With all the heterogeneity, how could we possibly treat these tumors? And the clue, I think, comes from the observation that despite the heterogeneity, the tumors follow conserved paths. Yes, we could look for epigenetic regulators that could push cells down one trajectory or another, and indeed that's something that we're doing. But another approach might be to target the cells that enter into these injured and fetal-like states, especially the fetal state, which is the entry point to non-canonical differentiation. And the way we've approached this is to look in organoids from metastatic colon cancer patients which retain these fetal programs. And so we hypothesize that the fetal programs, which are not expressed in most adult tissues, could represent exciting targets for therapy. And in this case, since many of these are intracellular, we've thought to look at the immunopeptidome, the proteins that are processed and presented on MHC class 1 on the cell surface of these cells in the organoids that provide an inexhaustible, expandable, and synchronized source of cells in the fetal state. So with Josh Lee, a very talented clinical fellow in the lab in collaboration with David Scheinberg, we've done immunopeptidomics on our organoids, focusing on tumors with HLA-A*02:01, which is the most common HLA haplotype in the Western populations. We've identified proteins that are selectively expressed only in the tumor organoids and gone through a rigorous screening funnel to make sure they're not expressed in normal colon organoids or other normal tissues. And indeed, through this sort of independent analysis, we again identify peptides that are unique to the fetal immunopeptidome, the fetal colon, that are associated with fetal Wnt signaling in early embryogenesis that are not expressed in normal adult tissues as our top candidates. And these are not expressed in the normal tissues. As you can see, they're expressed in the primary tumor and even more highly expressed in metastases. And now with these peptides in hand, what Josh has been doing is pulsing these peptides into patients' blood and looking to see whether there are tumor reactive T cells that we can pull out of patients' blood that can recognize these effectively foreign peptides, because the thymus, where T cells are selected, really only develops in the second trimester. And by that time, the expression of these peptides would have gone away. So there should, in principle, not be any thymic selection against T cells that can recognize these peptides. And indeed, that's what he finds in multiple donors. He is able to find donor T cells that can recognize these fetal peptides. And he's currently in the process of sequencing, cloning, and testing these TCR T cells with the goal, of course, of eventually taking this back into the clinic to try and treat our patients with advanced colon cancer. So in the interest of time, I'll stop there and thank, of course, our phenomenal lab, the people who did the work, Ellie Benitez, and Josh Lee, and Francesco Cambuli in my lab, Andrew Moorman in Donna Pear's lab, leading the computational analysis that I showed you, and David Scheinberg on the immunopeptidomics front. Thank you. Yeah.
Very interesting. Thank you. You picture this as a plasticity where these non-canonical states can be populated, I would assume, in a few cell divisions type rate and not a mutation or variant that then grows out. Is that the way you picture this unstable non-canonical state?
Yes, I think, yes. So I think it's hard to tease about whether it can happen within one cell cycle. It's easy to show that within a few cell cycles, this can definitely happen. And we do see evidence that the epigenetic state of these cells is different from the cells that are unable to undergo this plasticity. Is that answering your question?
When you say epigenetic states of these cells that undergo that plasticity, you're talking about chromatin accessibility and things like that.
Exactly, chromatin accessibility, histone marks. We're still in early days of trying to really understand exactly what is going on, but they are clearly, the cells that are capable of doing it have a different chromatin landscape than the ones that are not.
Right. Next question.
Sure.
So you see that SOX2 comes up, and is that something that is happening when you lose PROX1 normally or sporadically? Is that actually a predictive marker, or is it something that just turns on again with some probability but isn't deterministic? So I guess the dedifferentiation is creating, again, plasticity where the cells can then take on the identity of something else or very specifically something. And is that specific then to the types of metastasis and maybe even the regions that you observe?
Yeah, that's a great question. So we think that the dedifferentiation comes first and is necessary to enable the lineage switching to happen. And I guess the way we're seeing it, the question of how many lineages can you switch into is an important one. And I don't think we have a good answer for that yet. We see highly conserved, so squamous and neuroendocrine we see in pretty much every tumor we've looked at. The bone lineage is less common. We see more of this in the tumors that metastasize to the liver or the peritoneum, whereas in the lung, we see actually much more benign not even transdifferentiation, just differentiation down intestinal trajectories. So, there's clearly going to be local effects of the microenvironment, but also, maybe it's just easier for whatever epigenetic reason for these cells to enter into some lineages rather than others. Or there could be a selective advantage. All these questions remain unanswered.
Thank you for a great talk. So I'm just, this is definitely a brand new discovery. I try to associate with common subtype of the CRC that have like inflammatory microsatellite unstable, canonic, and the fibroblast triggers. I'm just wondering how this program you found can be mapped back to the traditional constance molecular subtypes.
Yeah, it's a good question. So in CRC, people have used bulk RNA sequencing to classify the tumors into 4 sort of consensus molecular subtypes. The vast majority of these non-canonical states are in CMS4, the most aggressive version, if you like. But what we're seeing is that CMS4 is a mixed bag of quite heterogeneous tumors. Whereas the CMS2, which is the more canonical, those tumors are the ones that even in our data set are more canonical.
Oh, thank you.
Okay. All right. Thank you, Emma. Right there.
All right, and that brings us to a break. We'll be back at 11 to start our next part of the session. Thank you.